everyone, welcome to today's webinar. My name is Melissa and I will be your host for today. So today's webinar we have Ola who's going to talk to us about using the MGnify API, and I'm going to hand over to her now to do that. So we're both going to turn our webcams off and we can get started.
Welcome everyone. Welcome everyone. My name is Ola Tarkowska. I work as a senior software engineer here in EBI, developing MGnify resource, which was previously known as EBI Metagenomics. And thank you for coming to webinar about analyzing and visualizing microbiome-derived datasets using the MGnify REST API. Today, I would like to show you how to access data from MGnify resource programmatically that allows you to perform more complex analysis and visualizations. I hope it will help you to understand how to access the data using the MGnify API, how to filter datasets by environmental metadata, how to write scripts to programmatically access the data to perform analysis using RStudio. First, I would like to say a few words about our MGnify website, about MGnify resource. MGnify allows retrieval of over 150,000 publicly available metagenomics, metatranscriptomics, amplicon, and assembly datasets sampled from diverse environments. This number is growing daily. MGnify offers an automated pipeline for the analysis and archiving of microbiome data to help determine the taxonomic diversity and functional and metabolic potential of environmental samples. Users can submit their own data for analysis or freely browse all of the analyzed public datasets held within our resource. In addition, users can request also analysis of any datasets which are held within the European Nucleotide Archives, which we then pull and analyze for you. So this functionality is available through our website. Once you go there, there is a request analysis section where you can do it. Our website is showing the analysis results, which is 100% driven by the API. So every data that you can find on the website can be accessed programmatically. So first of all, I would like to say what exactly API is. Application Programming Interface is a tool to access and process the data from the web resource, allowing data to be integrated for mapping, visualization, and other form of representation and analysis. APIs are used for data exchange or data mining. They allow to access data regardless of the programming language, so that you can write scripts in Python, R, or using other programming languages or tools. API is nothing else than like a power socket at home. It supplies home equipment with electricity. So basically, as long as the plugs fit into the socket, you can fetch the data. So as long as you use appropriate libraries to consume the API, you can do it programmatically. You can do it from using various programming languages. Here are a few examples of visualizations. So going through the from the left top corner, for example, you can download metadata for selected samples. Right top corner is a plot taxonomic abundance for selected environment, straight from the API. You can visualize graphical distribution of certain organisms from selected projects. You can also investigate correlations between metadata and gene functions. I will be showing you later on how to do it directly and showing you how to write the script in RStudio later in the webinar. So a couple of technical information about how to browse the data using the API. First, it's worth to start from our documentation to look there what kind of endpoints API offer, what kind of attributes you can get on any endpoint, what kind of filters you can apply. So everything is documented on the first URL. These URLs will be provided later after the webinar as a part of the summary. Most important is to know the base URL because this is your entry point to fetch the data. It's important to know that our API is hypermedia driven, that improves discoverability. It implements JSON API specification, defining formatted data structure, and provides schema using Open API specification that help us actually to automatically build the documentation. So this is our documentation. It offers comprehensive specification of collections, including filters and attributes, as I said before. So I will leave you this to examine after webinar. So this is mainly it in terms of the technical, the introduction and the technical part. So I will right now switch to web browser and show you how to access this data. Let me switch to our website. So this is MGnify website. On top menu, you can choose API. This will take you to API browser. So the base URL provides you with access to several resources, resource collections, such as, for example, studies, samples, analysis, experiment times, biomes. Let's click first on the studies. This took us to the study collection. It's a paginated list of all the publicly available studies held within a resource. So here we can see the pages you can browse through, and in API response, you have the same. You have, as I mentioned before, that's hypermedia driven, so you have a link constructed automatically for the first, last page, next, preview is not available on the first one. And in this response, you can also scroll down, just a second. Oh, technical. Oh, why can I not move this? Okay, this. Okay, so I can scroll down to the very bottom. On the very bottom, you have the metadata section of the API response, telling you about the number of pages and studies that are available. So here we can see over 2,300 public studies. So this response is the default format for the data exchange in API is known as JSON, which is a very compact and human-readable way of representing the data. So everyone probably came across JSON at least once in their life. So a MGnify REST API returns JSON. It's called JSON API's object formatted data structure that contains the resource type, which is telling you here what kind of object you get. Associated object identifier, which is our own accession. So MGnify resource offer two types of accessions. The first one is for studies, which contains MGYS prefix and the identification number, and this correlate with bioprojects or ENA accessions. And then we also accession analysis results. They start from MGYA, and they also have their own identification number. So in addition, we have type ID and list of attributes which are relevant to particular object, and relationships. And linking relationships allows also to construct complex queries once you would like to retrieve the data. So at the moment, we are on the studies endpoint. So for example, if you would like to access directly one of the study, let's not pick the random one. Let's try something which is known, like ocean sampling day. This is ERP 009703. So details about the single study can be retrieved by providing this unique identifier in a URL. And then we're retrieving just this study. In order to retrieve a list of samples contained within this study, we need to find in a relationship samples. So we have also a link. It's automatically generated for you as a part of the API response. So once you write the script, you don't need to worry about figuring out how to construct links. They are always there. So when we go to samples, we can see that there is 162 samples. So if we go to the very bottom again, to the metadata section, we have, yes, 162. This is also paginated. So in addition to samples, for example, if you would like to retrieve all the analysis in this study, we go to relationship and retrieve analysis performed on this study. So here we are basically calling studies endpoint. We're giving the identifier has changed while the URLs were generated, but any of them return the same data. So you can use any identifiers that basically match this study, and we are retrieving analysis. So in addition to this URL, you can parameterize it, and parameters can be added to this URL to help sort and filter returned data. So for example, we know that there were 162 samples, but we have over 600 analysis results, which obviously doesn't match well, but this is coming from the fact that this study was reanalyzed with the different pipelines. And you can actually compare within a study how tools affect analysis results as well. So it's always worth to keep in mind that you should always compare the same results from the same pipeline. So they can be filtered. Here is a filter section where you can actually construct the filters. How would you like to filter? So we can choose pipeline. I'm actually know that these results were these samples were analyzed with pipeline version 2 and version 4. So I will just pick 2 at the moment. Click submit. And this will give me much less because we have only 6 pages of the result for pipeline version 2. So I can do the same, pick here version 4, and this will generate 19 pages, which is over 400 results here. So this is pretty much the simple example how to browse our API through the web browser. Obviously, you want to do it through using scripts. This is only to show you how you can understand and figure out what kind of parameters and how this works. So right now I will switch to RStudio. Yes. So in this first example, I would like to show you that there is already third-party library written for R. It's called R-JSON API that allows you automatically to consume our API. So you don't need to write any library. You just need to fetch the results and process them for visualizations. If you're if you would like to do it in a different programming languages, there's the whole website. It's called JSONAPI.org, and on this website you can find pretty much packages in any programming languages, including Go, Java, Python, JavaScript, and any you would like to imagine. So in this script, I pre-installed the package, but if you want to do it at home, you will need to call install package and the name of this library. Then we are defining the library. We are defining the accession of the study. We would like to retrieve the data. So this is a very simple example showing how to make a connection to the API. So you acquire the connection to the base URL, which you provide here. Then on this connection, you are asking the router to give, the script is asking the router to give the samples for the study accession. So it will construct, the library will construct URL itself for you. And then we select columns which were fetched in the samples, combine them into the data frame, and save to a CSV file on the hard drive. So I will click source now. And this loaded the data. So I think I need to switch to, this one. And we have, okay. So now I need to switch again, sorry. So this is the result we achieved. So in this study, there were just two samples. We loaded accessions, longitude, latitude, sample name, geographical location, and environment and biome, which specify environment. So these samples were from two different environments. So let's come back to our studio. So there is another example, which will be a little bit more complicated because it iterates through the whole endpoint, through every samples, and apply filters. So in this, we follow the same strategy: create the connection, clean up the object, and define the data frame where we would like to store the results, and start from page 1. We will repeat until last page. We will ask router to create a request which will use biome endpoint on requesting all the aquatic samples. On the latitude greater than 70, experiment type is just metagenomic. They will be ordered by accession, and it will iterate for each page. Then we will select columns like accession, sample name, latitude, longitude, latitude, geographical location name, study, and biome. And then we will bind this together to save to the data frame. And once this loop finished, we will save this to the CSV file. So I will source this file again. It's retrieving each page separately. It's not very many pages, but you can apply any filter you like. So this is just an example of to match the Arctic Ocean, because it's latitude greater than 70. So when I open again the results in Excel, so we can see the results here. So we receive all the sample accessions, sample names, longitude, latitude for those samples, geographical locations, so these are Arctic Ocean, study accession, and biome. So these are all marines, some sediment. Yeah, there can be a mix of different samples because the probably longitude and we would need to provide longitude and latitude more accurate. I just took the one value and fetch everything what's available. So it's always worth to check whether the data was enough well filtered. So if I come back again to RStudio, we can change here actually. So we would like to get all the aquatic samples, but let's do it for ocean sampling day. So we will alter this query by adding study accession equals ERP009703. And so then we can remove the study because we know which study is it. It's just one. And let's source this again. It will overwrite. Let's save this to 2. Sorry. I will save and study. Oh, sorry, I misspelled accession. That's why it went wrong. So it's just one page. And if we open again in Excel, we can see just 3 samples. And they are definitely OSD because in a sample name you can see OSD. So they are marine samples. So there are various ways you can access this data, apply various filters. There are enough, it should be enough many endpoints to get the data you actually require. So now I will show you one of the last. This is the last example. It's a bit more complicated because it will. It shows you how to plot relative abundance of the taxonomic lineages retrieving taxonomic results straight from the API. In this exercise, in this example, you can see how to visualize the analysis results of the study where the DNA was extracted from subsea floor sediments and the domain-specific 16S ribosomal RNA gene primers were used to profile the archaea and bacterial taxonomic communities. So here in this script, we're preloading a couple of other libraries to do plotting, to plot chart, bar chart. We also use JSON library and our JSON API. So we are acquiring connection to the API. We specify accession for the selected study. In this script, you can also, we provide other accessions if you would like to try other type of examples. Then we fetch all the analysis for this study. So we call the router to give us all the analysis on the study's endpoints for the given accession. From this analysis, we load all the accessions. And then we loop through all these accessions to fetch taxonomic analysis. So we are calling another endpoint. And then we are constructing the hierarchy. We are retrieving the taxonomy hierarchy, and we only fetch phylum level. So this allows us to aggregate and display the taxonomic abundance. So I will just run this script. And here, first we can see that there are 3 results. Those 3 results look to target bacterial population, and 2 other were targeting the archaea. We can also see similarities and differences on the phylum level composition for each analysis. So these are these are mainly all the examples. If you would like to try, If you would like to try more examples, here are the URLs to, this is URL to our GitHub repo, where you can actually download all the examples I showed you today. There is much more. There is a whole Python section. You can get those results provided in Jupyter notebooks, so it should simplify to run and visualize straight out of the resource. There's also a Docker file helping you to run Jupyter Notebook if you're interested in. If you would like to contact us about any details, if you have any questions, feel free to do it directly via the contact URL. Your message will reach our help desk. Yeah, and I think this is mainly it. I would like to thank you, the whole team, for all the work on our resource, our collaborators from ENA, and our funders. That's it. I think there are questions. Yeah, we can move to questions.
Yes, thank you, Ola. We do have time for questions. If you could just move to the next slide. So if you do have a question, you can type that into the chat box. While you do that,
should I mute myself?
Yes, if you just move to the next slide.
Sorry.
So while you do that, a little bit about our webinar series. So this webinar was part of our regular Wednesday webinar series. It was in fact the last one for 2018, but we're going to be starting again in February, and there'll be more information about those webinars on our website, which you can see here soon. So do check back to see what is on there. If you're interested in accessing EBI resources programmatically, we do have a webinar series that we ran 2 years ago now that looked at a variety of different resources, and you can find that in Train Online. Finally, just a quick reminder to please fill in the survey that will pop up in your window after the webinar finishes. So let's have a look and see if there are questions.
So one of the questions is, will all the SRA dataset will also be available via MGnify? So if you would like anything to be analyzed, please fill in the form to request, and then we will analyze. We prioritize mainly private studies for analysis. So if you have any particular you would like to do it, please go to our website and let us know which one would you like.
Are there any other questions this afternoon? Okay, if there are no other questions today, I'd just like to thank you all for joining us. I hope you've got something out of this webinar, and hope to see you again in another webinar. Thanks, and enjoy the rest of your day.